Let's talk about how it's different with sex chromosomes. So a gene that's located on any sex chromosome is called a sex-linked gene. So when we say something is sex-linked, we're saying that it's physically linked. If it is an X-linked gene, then it is physically located on the X chromosome. And if it is a Y-linked gene, it is physically located on the Y chromosome. So sex-linked genes are physically located on sex chromosomes, not autosomes, and they can be either on the X or Y. So it used to be thought that all genes behaved the same way. And part of that is because plants don't have sex chromosomes, but animals largely do. And these experiments were originally done in the fruit fly. And what they found is that depending on which individual was the male, sometimes there were differences in the F1. And that didn't make sense. So, for example, in this situation, if we're crossing a male, a white-eyed male, to a red-eyed female, all of the progeny have red eyes. But now if we take these guys and cross them together, the white comes back. So how did that happen? So when they crossed the red-eyed males and females, they got a 3-to-1 ratio, which is what you would expect, except there was sex bias. Only males had white eyes. So what could possibly be going on here? The hypothesis was that the white eye trait was located on the X chromosome, and if it is, then that makes sense. So in this situation, we have the female having heterozygosity for the X, the male only has the dominant allele for the X, and this creates a situation where males can inherit the Y from the father and the recessive X from the mother, but females cannot inherit two of the recessive alleles. So they cannot have the white eye. So this creates a situation where we see sex bias. Anytime you see sex bias in a cross, your first assumption should be that this is a sex-linked trait. In the original cross, they had a white-eyed male. Let's say that we start with a white-eyed female. So that's going to be this. We cross her to a red-eyed male. We're going to get not all red eyes this time. Instead, we're going to get females that have all red eyes and males that have white eyes. So see, the F1 is different this time. Last time we did it the other way, we got all red. When we cross these two together, so here's our heterozygous female. Here's our white male. This is our F2 this time. We're going to get both females and males that have white eyes, which is not what we got last time. The sex of the parent matters. This also suggests a sex-linked trait. So when the male was the one with the white eyes, everyone was red, and then only males had white in the F2. When the female had white eyes, we got both red and white, but they were sex delineated in the F1, and that delineation went away in the F2. Okay, so who has the phenotype matters for this type of trait. And you're going to see sex bias in one of the generations at least. So in one direction, you're going to see it in F2. Here it is for F2. In the other direction, you're seeing it in F1. Here it is in the F1. That's your tip off, it's not autosomal. We know that the X chromosome has a lot of genes on it. We also know that it is associated with lots of genetic disorders, most of which appear in males. The reason for that is that males only have one X chromosome, so whatever they happen to carry is what they're going to show. In fact, a common example is color blindness, which is more typical in males. Females have to carry 2 copies, and red-green defects tend to be the most common. There are two different ways you can inherit X chromosome mutations, and they are the same ways that we think of for autosomal mutations. They can either be recessive or they can be dominant. In X-linked recessive individuals, let's say we're starting with an affected father. In this situation, we would expect the father to give the X to his daughters and not his sons. All of the daughters are going to receive a normal copy from their unaffected mother, and that will make his daughters carriers. The father does not give his X chromosomes to his sons. They only get their X chromosomes from their mothers. So they will not be carriers, nor will they have the phenotype. But how does this change if the trait is dominant? In this case, the father will give the dominant trait to his daughters, but not his sons. So now, instead of having carrier daughters, he will have affected daughters. This kind of sex bias, where the females are affected but the males are not, or vice versa, are again a suggestion that this is going to be a linked disease. If instead of being X linked, it were Y linked, how would that change who was affected? He's going to give the Y now to his sons, and all of his daughters would be unaffected. Now the sons would be affected. Right? So depending on what is going on with where this is located, is it recessive, dominant, or on the Y, will change the outcome. So most sex-linked traits in humans are going to be on the X chromosome. Most of them are due to recessive alleles, and that means that they are usually seen in men. So a male that receives a single X-linked trait from his mother is going to have that disorder, but because females have to get two, they tend to only be carriers. There are X-linked traits that are dominant. Females tend to have less of a phenotype. Let's think for a minute. Why would a female have less of a phenotype than the male in this case? We talked about in regulation of expression about something really big that happens to X chromosomes. In fact, most of the X chromosome is silenced during X inactivation. This means that with dominant X-linked traits, males do not silence this. It's going to be everywhere. But in females, depending on where the mutation is, some of the cells are going to be normal because they're going to be expressing this recessive X that does not have the dominant trait. Right? So remember that things like X inactivation play a role in determining how severe a phenotype might be. That's because of this whole dosage compensation system, where we're going to turn some of them into bar bodies. If we happen to silence it enough, we might not even see the phenotype, even though it is dominant. This is another example of how we can see things like changes in expressivity, where the degree of the phenotype is different across individuals. The timing of inactivation is also going to happen really early. So it's going to happen around the 8 to 10 cell stage during day 4. That means that that is where we're going to create our populations of cells that will or won't have this phenotype. And of course, the classic example that we discussed with things like calico cats also helps us understand how some places in the female body might express the mutation while others do not. Females with this kind of X inactivation are going to be mosaics. So if they're carriers of recessive or dominant traits, they will express them in some cells but not others. This is also why we see this idea of the manifesting heterozygote. Most of the time when we're thinking about heterozygosity like this, we would expect that this is either going to be, dominant, whatever the R is, we'll see the R phenotype, or we'll see some kind of intermediate phenotype. However, because some of the cells are going to express this recessive one, we'll sometimes see carrier females for X-linked traits having the phenotype if they happen to get unlucky with where extivation happens. Right? So not only do they have less of a phenotype for dominant traits, sometimes they'll have more phenotype for recessive ones. This is also why it's much harder for women to get diagnoses for these types of traits. Because not all of their cells are affected, they might only have the phenotype in some of their organs, but not others, or some areas on their skin, but not others, whereas the male will have it everywhere. Another example of this, which makes it clearer in humans, is Blaschko lines. Blaschko lines are specific areas where development happens and cells migrate during development. These areas are consistent and they follow very specific patterns, and every human being has them. But you can't see them most of the time because your skin is largely all the same color. However, small mutations in pigmentation in skin can sometimes show you where the lines are. X inactivation in heterozygous women can have different phenotypes depending on when it happens before migration and which cells are affected. This is why things like the loss of sweat glands, which can occur through one specific mutation, doesn't happen across the entire body, but instead follows the Blaschko lines, depending on which cells were initially affected. So I'd like for you to understand about this is that sex-linked traits are inherited differently. For all of our problems that we'll be solving, you can assume that X inactivation is not involved. But when thinking about diagnosing someone or working with an individual with an X linked trait, understanding that sometimes men and women are affected by that trait differently because of how dosage compensation or expression works in those individuals can help you make better diagnoses. Now, not all X linked traits are going to behave this way. Remember that there are genes on the X that are on all the time. So while some X-linked traits will see this type of inactivation, not all of them do. The most important genes that are expressed off the X are expressed off of both sides in women to make sure that enough is produced. So some genes, this is not going to show up for. Not all heterozygous traits will behave this way. All right, so how do we identify this? We're looking for biases in sex. So anytime that we see there's sex bias, anytime that we see that who has the trait matters. So we're going to see transmission from fathers to daughters and from women to both of their children. And we might see differences in expression between males and females because of how the X's are used. Right? Those are all going to be tip-offs for this being an X-linked trait.